Hello students, welcome to Ural NEET classes. Today let's see principles of inheritance topic containing numerical problems. And this topic was divided into three subtopics for our easy discussion. So like Mendelian inheritance, post-Mendelian inheritance and human genetic problems. Got it? So in this video we will discuss Mendelian inheritance numerical problems. The first one. Out of a population of 1600 individuals in F2 generation of a cross between yellow round and green wrinkled seeds of a pea plant, what would be the number of green wrinkled seeds? See, every time when you are solving the questions, you should find the keyword. So the keyword for this question is we should identify or we should find the number for green wrinkled seeds. So the given options are 100, 200, 50, 25. Got it? So if you observe properly, it looks like a dihybrid cross, because here color of the seed is taken into consideration as well as shape of the seed also taken into consideration. So it is in a dihybrid cross, and we know the dihybrid cross ratio, the phenotypic ratio is 9:3:3:1. Got it? So directly we can identify the number of the given criteria, that is how directly we can find green wrinkled seeds number is how much by using this ratio. Before that, you should have an idea about what is 9, what is 3, and what is this 3 and what is 1. Got it? So if you see in your NCERT, typically you have a rhomboid shaped diagram will be the NCERT. Okay, ma? So when you observe that carefully, you will get these ratios. So if you observe, the 9 are nothing but yellow round seeds. The first 3 are nothing but yellow wrinkled. The second 3 are nothing but green round. The last one is nothing but green wrinkled. So as for the question, what we want, green wrinkled. So the formula we are going to apply or use is number of phenotypes. If you want to know in the given number of a population, what number of phenotypes is required, means the number of phenotypes is equal to total number of population into phenotypic ratio by total number of phenotypic individuals. So in this dihybrid cross, we have got how many? 1600 individuals, and we want to know green wrinkled seeds. In that we want to know one ratio. Got it, ma? So if you observe yellow round 1600 into 9 by 16, we get 900. Yellow wrinkled 1600 into 3 by 16, we get 300. And green round is equal to 1600 into 3 by 6 is equal to 300. Green wrinkle is equal to 1600 by 16 is equal to 100. So why we are taking 9:3:3:1, that is as per the phenotypic ratio in the Mendelian cross. Got it, ma? So here if the question required is green wrinkled seeds, those are nothing but 100 in number. The answer is 1, that is nothing but 100. Is that clear, ma? So this totally, if you remember this formula, easily you can do it, ma. Got it? Second thing, you should remember this ratio as well. Clear? Okay, ma? So let's see the next problem. Same like a previous question, ma, just I changed only the numerical of individuals in a population. So you can try this one. So please grab a paper and pen, ma. Please try to solve everyone with the previous things. Just pause the video of a previous one and try to solve it, ma. It's almost same like that only. Got it? So same thing, ma. The dihybrid phenotypic ratio is 9:3:3:1. Here we are what we want, ma. Same like previous question, green wrinkle seeds only. So directly 400 into 1 by 16 is equal to 25. So the answer is what, ma? 25. Got it? So you can change the 400 to 200 or 1800. You take any number, ma, and you can solve it for your perfection. So please do it, ma, by changing the number of individual population. Let's move on to the next question, ma. Question number 3. The parental genotype is capital A, small b, small b, capital C, small d, capital E, small e. Crossed with another parent genotype is capital A, small b, small c, capital D, small e, small e, and produced offsprings. What is the percentage of offsprings with the genotype of capital A, small b, capital C, capital D, small e, small e. So it looks like a penta hybrid cross. That means 5 characters we are observing at a time in this one. If you do regular Punnett square method to this one, it's going to be full time consuming process. So for this purpose, what we are going to do, individual genotype we are going to identify. That means we are doing individual monohybrid cross. How? Like this. Whatever the blue boxes you are seeing on the screen, that is called as a monohybrid cross. So the two parents genotype gametes we are taking and we are going to cross, and we observe what is required in the offspring. So if you observe in the first case, the offspring requires the offspring contain heterozygous capital A, small a. Got it? So when the heterozygous parents were crossed, so in this case what happened, out of 4 offsprings, we got 2 offsprings with the desired genotype, that is capital A, small a. So those are 2 in number. So out of 4, 2 are there, so it will become fraction of 1 by 2. The same way we will do for the next monohybrid cross, small b of one parent cross with the small b of another parent. But what we desired in the next offspring is small b as well. So all will be the 4 all the offsprings will be the recessive homozygous only. So it become 4 by 4 is equal to 1 In the same way, third monohybrid cross we are going to do between capital C and small c of the both the parents, and we take the gametes. So we require in the offspring is a heterozygous condition, capital C, small c. So those individuals are like 4 by 4 is equal to 1, we get the fraction. Next monohybrid cross for the character is small d crossed with the capital D, small d. So what is the desired we require? Capital D, small d in the offspring. Those are 2 in number. So 2 by 4 is equal to 1 by 2 fraction, we got it. So the last character, one parent has a capital E, small e, then crossed with the small e. And what we require is in the offspring is a recessive character, that is small e. So those are of 2 individuals out of 4. So it's going to be 2 by 4 is equal to 1 by 2. So in this, what we should do means, after doing this individual monohybrid crosses, whatever the fraction, whatever the number we got it, we have to multiply them. So what is going to come here? 1 by 2 into 1 into 1 into 1 by 2 into 1 by 2. So we'll get what 1 by 8. So which is equal to 12.5%. So the answer is what? 12.5. Is that clear So like this, let them give any number of characters crossed. So simply by doing the monohybrid cross, you can do it. It will take a little time, but still easily you can do it. Got it? If fourth question, if 2816 seeds is obtained in a Mendelian dihybrid cross performed for the shape and color of seeds, then how many seeds would be of pure line? So remember here pure lines means generally every student will think that one, that 2 extreme ends, either homozygous dominant or homozygous recessive things. Got it? But the twist in this question is, even though we are comparing 2 characters at a time in an individual, if they have the alleles of same type, still we call them as homozygous individuals. For example, you can see here this capital R, capital Y is a purely homozygous condition, and small r, small y, this is also what homozygous individual. These both are. This generally we think it. What is the remaining 2 capital R, small y. Here capital R what These 2 are also homozygous conditions. The 2 alleles are homozygous conditions. Small y also in the homozygous condition. Even though they are present in a single individual, but we should observe the character containing 2 alleles in what type of status. Here the yellow color thing, got it? The color of the seed is small y. Got it And here capital Y, and the shape is small r, and the shape is capital. So these last 2 also called as homozygous individuals. This is the logic you should apply then only you can get the answer. Got it? So now let's see the remaining steps of this one. So capital R, capital Y. So they are how many individuals in total dihybrid cross here? Like this we can see only 1 individual out of 16. So 1 by 16 into 2816 is equal to how much, ma? 176 we get it. In the same way for homozygous recessive also will get 176 individuals. Got it? And even to your surprise, this capital R, small y individual also will be 1 in out of 16 only. Got it? So in this case also we get 176. Same thing also apply for the small r, capital Y. This is the only one individual present out of 16. So here again we should multiply 1 by 16 into 2816, we get 176. So the question is how many? That means we should club everything, then we got the total thing. So total 704 individuals we are going to get it who are homozygous in condition or pure line. Got it, ma? So answer should be the C. Next question. Fifth question, ma. The percentage of heterozygous individuals obtained in F2 generation from selfing the plant with the genotype capital R, small r would be. So the given options are 24, 50, 75, 100. So when you do the Punnett square, like it comes, because it's a selfing, that means we should take both male and female as capital R small r and capital R small r. So this is the respective gamete. So we are going to get it's a mono, it's a what, ma? Mono hybrid cross. So if you in this one, if you observe the heterozygous individual capital R small r of 2 individuals are going to come. So that means what, ma? It's going to be 50%. Got it? Half, because out of 4, 2 means 50%, ma. Got it, ma? So this is a simple question, yet I have taken a sample questions for the sake of our discussion. So please remember this one, ma. Now let's move to the next question. Sixth one. When a cross is made between tall plant with yellow seeds, that is capital T small t, capital Y small y, and dwarf with green seed, what proportion of tall with green seed phenotype in offsprings could be expected? So whom we need to expect, ma? Tall with the green. Got it? So if you observe, this is the table we are going to observe. We know that when the dihybrid cross, we are going to get 16 individuals. So when you see that one, in the above cross, we have seen a heterozygous individual is crossing with the recessive individual. So when you take the gametes for capital T, capital Y, small y, we are going to get 4 different types of gametes. Got it? So when you take a dwarf with a green, in that we are going to get only one type of gametes. So when you do the cross, the expected proportion of tall green, ma. What do we want, ma, here? Tall green. Tall means what, ma? Capital T, you can take it. And green means what, ma? Small y, small y. So when you see the second column, individuals are this category, tall with the green, tall with the green. So how many individuals are there? 4 individuals out of how many? 16. So it is equal to nothing but, ma? 1 by 4. 1 by 4 is nothing but 25%. Got it, ma? So 25, 25, 50, 50 plus 25, 75 plus 25, 100. So out of 100 percentage, 25%, nothing but 4 individuals are of tall and green. Got it, ma? So this is how we have to solve this one, ma. So we move to the next question. 7th one. In Mendel's dihybrid cross for seed shape and seed color, the F2 progeny produced with the genotype capital R small r, capital Y small y, small r, capital Y small y, capital R small r, y, small y, small r, small y, small y are respectively. So simple, ma. The question itself is given, it's a dihybrid cross. Students, what you should do is you should remember these combinations of these alleles. Of course, everyone remember the table of 16. The first will be capital R, capital Y, capital Y. This extreme end you are going to remember is small r, small y, small y. The in between things only the confusion part. So I'm going to teach you simple logic, ma. Observe. So if you observe the first 4 are this capital R, capital Y, this RR in homozygous condition, YY also in homozygous condition. Here also second one, small r, homozygous condition, small y are also homozygous condition. But come to the third one, you can see here capital R, homozygous condition, and the Y's are small y in homozygous condition. Got it? In the first 2 cases, either 2 characters related alleles are in total dominant or total recessive. But in the third type, half are dominant, half are recessive, but both are homozygous in condition. In the same way, small r, capital Y. So here also two are two are recessive, two are dominant, but both are individual are like a homozygous condition. So these kind of genotype ratio will be are having only one individuals. That means 4 are going to present like this. Got it? And the next 4 are going to be purely heterozygous condition. One capital, one small, one capital, one small. Got it? Now 4 and 4, 8. So 8 easily can rule out. The simple logic is another 8 are nothing but all possible combinations of one will be homozygous condition, one will be heterozygous condition. These we can see 4 types, but each 4 type will contain 2 numbers. So total 8 individuals. So it is going to be what, 8 by 16. So total 16 by 16 we got, So as per the question, if you pick up them with the bold letters, you will get the ratio of 4:2:2:2:1. 2:2:2:1, 4:2:2:2:1. Got it? So let's see the next question. 8th one. In the monohybrid cross, capital T small t crossed with small t, the phenotypic ratio of the progeny would be, okay, so it looks like a typical test cross. So when you take the gametes and when you cross it, so what is asking, phenotypic ratio of progeny would be. So if you see the phenotype, we get one tall, 2 are heterozygous tall and 2 are homozygous or recessive. That means a dwarf. So we got 2 tall and 2 dwarfs. So nothing but those are going to be 2:2 ratio, nothing but 1:1 ratio. So in the given option we have 1:1 ratio. So the answer should be the 1. Got it, Okay. So with this, what is my intention of doing this video is just for the sake of your revision, Second thing, some important tips I want to tell for you this topic is please never ever do the crosses with your eyes. Please take a piece of paper and a pen, roughly solve it, then you will get a full clarity. Got it, So please never ever do with the eyes. Never ever count in the mouth. Cross check with by drawing the Punnett square. It takes, if you practice properly, it won't take less than 15 seconds, Fadafat you can do it and you can solve it. Got it, Okay then. So we will see in the next slide, in the next video regarding the post-mental inheritance and human genetical disorders things. So please like to reach more students like you, and please do the comment what you require, and share to your friends if you like this content. And to stay with me, please subscribe. Thank you.